Hello, everybody, and welcome to our latest event in the Edmund and Sylvia Morris Lecture Series. My name is Sarah Marshall, and I'm the director of Kent Memorial Library here in town. We are so glad that you could join us today for what will be a fascinating conversation about timely topics in medicine with Dr. Harold Varmus. But first, a few words about the Morrises. Edmund and Sylvia Morris were renowned biographers and accomplished writers. Edmund won a Pulitzer Prize for his biography of Theodore Roosevelt and followed it with books on Reagan, Beethoven, and Edison. Sylvia was also an acclaimed biographer, penning works on Edith Roosevelt and Clare Boothe Luce. She was a sought-after lecturer, too, and gave talks at the Library of Congress and the University of Delaware, among others. Both were deep-thinking scholars and library lovers, and when they passed, our friends, Donna and Ben Rosen, reached out with an opportunity to design and present a speaker series honoring the legacy of these two esteemed members of our literary community and our community in Kent. Our goal with the Morris Lecture Series is to tackle big issues and start important conversations, and we hope you will be inspired by these events to think about the challenges we face as a town and as a nation. And now here's Brittany McAllister, Director of Adult Programs and Special Events.
Thank you, Sarah. I just wanted to say thank you, welcome. This is going to be a wonderful program. And towards the end, our wonderful moderator, Bert, is going to signal to the audience that it's time for Q&A. A volunteer and I will have microphones, so if you do have a question pertaining to the conversation, raise your hand and we'll bring over a microphone for you to speak in. That way we can have everything on the recording for later. And if you do have some time before your dinner plans this evening, we will have a wine reception. We're hoping the rain holds out and we can have it outside, but if not, we'll just bring in the tables inside and we can mingle and debrief as a community and talk about what we just listened to. And it is my pleasure to introduce tonight's moderator. Bert Wells is a lawyer and a former mathematician who has had a lifelong love of science. He earned his doctorate in mathematics at Oxford University as a Marshall Scholar, and after a short career as an academic research mathematician, he switched to law, graduating from Yale Law School. Bert is a partner in the New York City office of Covington and Burling, a global law firm where he practices corporate insurance recovery law, enabling his clients to collect insurance for calamities such as floods, cyber attacks, class action litigation, and most recently, COVID-19. So thank you, Bert.
Thank you for that introduction, Brittany. It's my privilege to introduce Dr. Harold Varmus, who's both a legendary biomedical scientist and a humanist, both a thought leader and an institutional leader. He's the ideal person to hear from about the future of medicine. Harold's college and first graduate years were devoted to the study of English literature, but a switch to medical school and biomedical research soon led to important achievements, including the profound discovery of the basis of a new paradigm for the origin of cancer. His work was recognized with the Nobel Prize in Physiology or Medicine in 1989, besides many other notable prizes. Harold was never the sort of scientist to believe that his responsibility ended at the laboratory doors, however, and he went on to hold a series of exceptionally important leadership positions in the world of medical research. It's a dizzying list, but I'd like to share some highlights. For 6 years, in the decade of the 90s, he was director of the National Institutes of Health, the federal agency that conducts and funds biomedical research, successfully persuading Congress to increase the NIH budget dramatically over those years. For 10 years, the decade of the 20-aughts, he was the president and CEO of Memorial Sloan Kettering Cancer Center in NYC, which of course is a globally famous cancer research and treatment hospital. For 5 years, from 2010 to 2015, he was director of the National Cancer Institute, the Cancer Focus Sub-Institute of the NIH, where, as I mentioned, he had previously served as overall director before. Harold is now at Weill Cornell in New York City, where he runs a research lab and is professor of medicine. And he also manages to sustain an astonishing schedule of other important commitments. So the format today, we're going to have 30 to 40 minutes of discussion. There'll be 15 or 20 minutes of time for questions, and then, as Brittany mentioned, there'll be a reception right after. So let's jump right into it. And touching on the science, which has been...
I thought that was the end, Bert. That was good so far.
That's good enough, okay. Let's take advantage of the remaining 55 minutes of our time and see if we can talk about some interesting things here. So Harold, how do you, as a longtime cancer researcher, define cancer, or what characterizes it for you?
Good. Before I get into that thorny question, though, let me just say a word about my gratitude to all of you coming out on the weekend to talk about these issues, to the Rosens who have brought us back together to one of our favorite towns, at least in Connecticut, to have happy memories of Kent Presents as we roll past the Kent School and see some of the denizens of Kent coming out. And I'm particularly pleased to be doing this in honor of the Morrises, whom I'd only met once, but whose scholarship I've always admired. Cancer. So cancer is a complex thing that we respond to in a variety of ways. I think for many, the first notion of what a cancer is just results from hearing about a friend or a relative who was struck down by this disease. That sounds dreadful. When I was brought up, the word was rarely used, even though my father was a physician. It still was a dreaded term that one had to decide whether or not to mention. But for me as a scientist, cancer represents an intriguing phenomenon because it represents the misbehavior of cells. A cell exaggerating certain elements of its normal behavior, growth, invasion, change in the way it appears, the kinds of things it does metabolically or biochemically, in a way that is very difficult at first glance to understand. Why would a cell that is sitting happily in a tissue or sitting happily in a tissue culture dish suddenly misbehave? And when I began working on cancer, hate to say it, over 50 years ago, the mystery was what triggered that change. And there were many ideas in the intellectual environment, but the one that had the most resonance and the most preliminary evidence in favor of it was the idea that there were changes in our genes and that those changes somehow instructed cells to behave abnormally. Instead of ceasing growth when the tissue was fully formed, the cell keeps growing. Instead of obeying the normal boundaries between where a lung cell should be and a muscle cell should be, the cell invades. And in those days, although we knew that genes existed and had some conceptual notion of what a gene might be, we didn't really have our fingers on genes the way we do now. Now, all of you in the audience probably assume that you can take the genes in any individual, determine the precise set of instructions by doing DNA sequencing, a term that rattles off the tongues of non-scientists, but a concept that wasn't even really possible, at least not technically possible, 50 years ago. What we were able to learn by using viruses that cause cancers in animals is that, yes, genes can instruct cells to change, that we can get our hands on these genes initially by using the viruses that cause cancer, because viruses are very simple genetically, having just a few genes as opposed to the tens of thousands of genes that animals like ourselves have, and that changes in those genes and variations in the genes that we inherit, but more importantly, changes in genes that occur during life play a major role. And ever since that concept became increasingly likely, now accepted as biblical truth, maybe I shouldn't say it that way, but was accepted as highly supported evidence. It's been the job that I and many of my colleagues have tried to undertake to try to figure out exactly which genes are responsible, what those genes do when they become abnormal, and what we can do as oncologists or therapists to try to either change the genes, interrupt the effects they're having on cell behavior, or get rid of the cell that displays these abnormalities.
So that's great. That's a great starting point. So as we start to think about treatments, of course, cancer has been treated with varying degrees of success for decades, but how does this vision of cancer as being inherent in something about our genes that can change and cause cancer, how does that inform treatment? How were traditional treatments really responsive to that vision of cancer? Did they respond to something different? And how has the world of treatment changed in response to that view?
The triad of treatments that we're all familiar with are still extremely useful and sometimes curative: surgery, radiation, chemotherapy. But each of those, while often effective and often not, don't rely on a detailed knowledge of how cancer works. So you have an abnormal set of cells we call a cancer. It's a group of cells behaving badly. We take those cells out, and if there are very few or no cells having escaped into the non-operated area of the body, you can cure somebody or certainly slow the progression of the disease. Or if you take advantage of one attribute of cancer that is probably preeminent, namely the tendency of cancer cells to grow when they should not be growing, you can use toxic chemicals or radiation to kill cells that are growing excessively and have a bigger effect on growing cells, the cancer cells, than on the normal cells. But what has happened over the last several years, perhaps 2 decades, is 3 approaches that I think are, well, 2 of which I know are effective in many circumstances, and 1 which I predict will be increasingly effective. The first is to know exactly which genes are abnormal and to try to develop drugs that inactivate those acquired excessive activities of certain cancer genes.
Is that what is called targeted
So
therapy?
that's what we call targeted therapy, where you're targeting a drug specifically to a certain gene. And I mean, just at the risk of going beyond the bounds of Sunday discourse to the community, suggests that one way in which a gene could contribute to a cancer is by making an enzyme that is much more active than an enzyme would normally be, and that enzyme plays some role in growth control. If you had a chemical, a drug, that blocked the activity of that enzyme, you might interfere with the ability of that enzyme to make a cell grow like a cancer cell. And that, in fact, is the basis for many of, actually most of the so-called targeted therapies that are in use today. And it's effective, but it's effective only to a limited degree because cancer cells, like other cells, like the viruses we're most familiar with today, the coronaviruses that are causing the pandemic we're living through, cells that are cancerous evolve genetically, and they can evolve to evade the beneficial effects of these targeted drugs. The second thing that has come about is a detailed appreciation of how the immune system can recognize a cancer cell. For many years, I grew up in an environment in science in which it was felt that the idea that an immune system could eradicate a cancer was mainly disputed. Much evidence suggested that the immune system simply didn't have the chops that allowed it to eliminate a cancer cell. But we began to learn more through a detailed understanding of what the immune system consisted of, and learned that there were brakes on the immune system as well as accelerators, and that it was possible to interfere with the brakes on the immune system and basically unleash the immune system to recognize a cancer cell, which is, for the most part, very much like our normal cells, but has some abnormalities that can be addressed. Exactly what those abnormalities are is still mysterious, but the fact that we understand the immune system so much better, we now have immune therapies. They don't work for everybody. They don't work in all diseases. But I, and I suspect many of you, have friends who are alive today because of the new immunotherapies. The third area, which I see is one that's emerging quite quickly in scientific work, is to understand what some of us call cancer cell dependencies. Yes, cancer cells are motivated by the mutations they've acquired. They can be seen by the immune system. But in addition, cancer cells depend on a different set of genetic factors than normal cells. And that widens the repertoire of possible targets. That if a cancer cell is dependent on gene X and the normal cell is not, if we can kill gene X in the...
So the metabolism is relying on different...
Metabolism would be one attribute. I'm trying to avoid these physiological labels because part of it's about metabolism, part of it's about blood vessels, part of it's about other ways the cell has to regulate itself. Some of it actually gets down to a genetic level we don't want to get into here, but it suggests that there are components of a cell which are not themselves mutated, which become dependencies, become building blocks for the continued health of a cell that has also the mutations that render that cell cancerous. So it's a new appreciation that it's not just the mutations that are driving a cancer, but the attributes of a cell that allow a cancer cell to survive that could be somewhat more complex in the general gestalt here, but so it's not as simple to grasp.
So you describe it as taking advantage of cancer cell dependencies that are distinctive to the cancer cell. Is there a name for that kind of category or that approach?
These drugs are not yet in use, but one way to think about it is that a cancer cell has a special need for additional blood vessels that normally wouldn't appear. And we do have some drugs that work to prevent the development of new blood vessels that would feed a cancer. Cancer has a big appetite, and that appetite is for example, displayed because cancer cells take up a lot of glucose, but they need certain increase in nutrients, they need additional oxygen, and they need blood vessels that supply those things to the bloodstream. So those are dependencies that cancer cells have that normal cells don't have. This notion of the dependency and the ravenous quality of cancer cells is the grounds for a new book by a man named Sam Apple that just appeared called Ravenous. And it's about an interesting German biochemist who won a Nobel Prize in 1931 for his work on how sugar is metabolized. And then he evolved a thesis that cancer was actually the result of the altered metabolism of cancer cells. Now that's not quite right, but the biochemist in question, Otto Warburg, has had that notion come back into favor as we realize that the metabolism of cancer cells is, as he said, really fundamentally different. Warburg also was a half-Jewish man with an arrogant personality, very demanding, living openly with his male lab assistant, and spent all of World War II working pretty happily and affluently with the daily horseback ride and a huge renovated lab, despite his Jewish origins. And this new book called Ravenous, I recommend to people who are interested in this topic.
Sounds fascinating. So just going back to those categories, targeted therapy's been around for a couple of decades. Mm-hmm. There's still more mileage to get out of targeted therapies? There
are many, many biotech companies that have built their fortunes and are still building them on the notion of targeted therapies. And, I mentioned one of the greatest shortcoming here. First of all, they don't work for everybody. Secondly, resistance to drugs almost always emerges. But we haven't yet succeeded in doing things that I think the audience will recognize as a way to try to make targeted therapies stronger. After all, everyone knows about treatment of HIV with so-called antiretroviral therapies. And when those were first introduced, the principle really very much the same. The antiretroviral therapies are targeted therapies. They're targeted against the few genes that HIV uses to replicate. And when we began those therapies with a single drug against one of the viral genes, they weren't very good because the virus evolves and develops drug resistance. But now we know these therapies are very successful because we use multiple drugs at a time. And it's very hard for a virus to develop resistance to multiple drugs. So I think that's a place where we simply haven't yet developed a sufficiently coherent notion of how genes work together to cause cancer. I've emphasized mutations. What I haven't emphasized is that virtually all cancers are the result not of a single mutation, but multiple mutations, multiple variations in genes. So there are actually multiple targets. And the challenge is to, first of all, know which targeted therapy should be put together to make a drug combination. Secondly, to get the drug companies that have made these different drugs to put their drugs together and work in a fashion that promotes multi-targeted therapy, and then to deal with a remarkable complexity of disease types that have become much. It's not simply enough to say, my friend has lung cancer. We want to know what kind of lung cancer and which mutations are required for that disease. Lung cancer.
Right.
And the further complication, and my wife is probably thinking you should shut up and let Bert ask the questions, but the final complication is that everybody is inherently different. So the mutations that occur that push a normal cell to a cancerous state are being made on top of the inherited variations we have.
The unique genetic makeup of the patient.
Yeah.
So the future, just to sum up, would you say the future of cancer treatment is still very much in development, hopeful, lots of potential, plenty of reason to continue to invest in biomedical research and the development of new drugs?
I haven't closed my lab.
So we've talked a lot about cancer treatment. Just briefly, could you say a few words about the future of detection and prevention?
Yeah, so I'm glad you brought this up because people do tend to focus on therapy, and there's a good reason for that. We have friends who have cancer, we want to know how to treat them, we're concerned about getting it ourselves. But the detection issue and the prevention issues are really important. And one of the things, you were a lawyer interested in risk, and one of the things that we are getting better at, but still aren't great at, is deciding who is at risk. And we know that some risk is easy to define. Smoking is a risk factor. We now know that increasingly obesity is a risk factor. And we don't fully understand that, but the epidemiological data are now very strong. But we know less than we should know about inherited risk factors. We know about certain genes. I think everybody in this audience probably knows about the so-called BRCA or BRCA1 and 2 genes that strongly predispose to breast and ovarian cancer. They also predispose to other types of cancer, but at a much lower risk. And what I think we are missing is a clear definition of the ancestral factors, the things we inherit from our relatives that may place us at very modest but significant risk of many other cancers. And
so you would say risk and predisposition are basis for monitoring or more careful
It might well
attention.
be, yes. And it's also a way to understand cancers because those risk factors are themselves contributors to the behavior of a cancer cell. So one of the things that I'm going to preach for just a moment, we've tended to look at these genetic risk factors almost entirely in so-called white or Caucasian European descent families. And one of the things that I've been heavily engaged in for the last couple of years is trying to make use of the genetic diversity of New York through the work that I do at the New York Genome Center to have a deeper appreciation of the categories of genetic ancestral factors that might be more understandable by looking at a wider variety of our human species, Asian, South and East Asian, South American,
sounds very important,
African.
Scientifically important, yeah.
Scientifically important. There's a tremendous amount of diversity, and we tend to focus on a fairly modest subset of even the European population.
Right. Well, look, I would love to continue to talk with you about cancer for all afternoon, but let's switch gears and talk about COVID-19,
Sure.
which is certainly the issue of the day. So just put this horrible epidemic into proportion. Where does COVID-19 fit into the list of major causes of death and injury, like over the last year and a half?
Well, it's the explanation for about a 20 to 30% increase in mortality. So it's very substantial. And it's reduced the life expectancy in many countries. Of course, the distribution of those factors is very uneven. To everyone's surprise, the effects have been major in the U.S. The U.S. was considered several years ago to be one of the best prepared countries for a pandemic, but it turns out our public health system was in terrible shape. We hadn't given enough attention to the way in which we do prevention and respond to public health emergencies in this country. And we had a period of, frankly, poor leadership, poor political leadership during the first years of the pandemic. But and then there are a lot of other reasons why the disease has not been as common in some countries as others, and every case is different, having to do with travel patterns and the way people live. So when we look across the entire globe, we see that the mortality rates have been rather low in some other countries, even countries that are extremely poor. That is not a source of comfort, because the thing that is most alarming to me at the moment, as someone who's interested in public health, is that we are sitting on a powder keg here. We have the resources and the manufacturing know-how to provide vaccine to everybody.
To stop this, but what are these continuing dangers? I mean, that's what I want to get to.
So the danger is that we have large numbers of people who have not been vaccinated, have never seen this virus, who are likely to be the vehicles in which the virus grows, generates the variants that could not only transmit more efficiently, as we've seen already with the Delta variant and the Alpha variant, but could be unable to respond to the immune response that the vaccine induces. So my fear as a medical scientist, not actually active in this research area, but knowing enough virology to have concerns about it, see that we have left this huge population, several billion people who are not prepared to resist infection when it occurs, and we don't get vaccine to these people soon, we're going to have vaccine variants that are worse than
we're leaving the kettle on
we're
the
living
boil.
with now.
Yeah.
So,
is it just luck that the existing vaccines seem to protect against the Delta variant, or is it in fact?
It's a little hard to say.
broad?
I mean, it's not just luck that we have these vaccines. People seem to think that we were lucky to have made all these original investments even before we had the vaccine with what we call pre-production purchasing agreements. No, we have these vaccines because for the last 20 years, advanced biological science has produced methods like the use of RNA, which is very famous, but also the production of false viruses, pseudoviruses, we sometimes call them, collections of viral proteins that can very efficiently be assembled and can be used to vaccinate. And all those things have worked in an incredibly short time and very effectively. Now, the fact that almost all these vaccines generate we call a robust immune response is fortunate in some way and couldn't have been so easily predicted. But the focus has always been on the spike protein. The spike protein is that protein you see on the surface of this studded particle that I think probably everybody knows is the key that fits into the lock in our cells and allows the virus to gain entry to the cells. That is the target for this immune response. And because the spike protein is so essential to the life cycle of the virus, maybe at one level it's not that surprising. It all sort of makes sense. But it could easily have been the case that the part of the spike protein required for it to usher a virus particle into a cell would not have been such an attractive target for the immune system. The fact that it has helped us a lot.
We're fortunate in that regard. So just to be devil's advocate for a second, is there any chance that this disease could just run its course and effectively vanish? And now I'm thinking of the 1918 flu, for example, which, in the absence of vaccines, had a ghastly toll in terms of death and morbidity, but it did run its course and disappear. Is there any chance of that happening?
It's very difficult to exclude that, but all the evidence so far suggests that's not going to happen.
Is it a different disease, really?
Well, it's a different disease in the sense that the virus is in a different category.
Coronavirus instead of influenza.
These viruses have some similarity. They both have their genes in the form of RNA rather than DNA, but the genetic complexion, the genetic makeup of these viruses is very different. I don't think anybody can tell you exactly why the influenza epidemic faded away in 1918. But you're right, we didn't have an effective vaccine, yet it happened. One of the factors that has to be acknowledged is that we move much more rapidly today, and we move at a much higher frequency.
The mixing of people.
And mixing of people. So one of the things your question raises is how do we monitor what's going on more effectively? And how do we prepare for the next pandemic, whether it's a coronavirus pandemic, which is not crazy to imagine. After all, we've had MERS, SARS-1, so it's not inconceivable the next pandemic will be a coronavirus pandemic, but could be a totally different kind of organism as well. And I think,
well, can we wait 100 years for that next pandemic, or?
Well, we've already had, if you count Zika as well and influenza, we've had a lot of epidemics or pandemics in the last 20 years. I don't think it'd be very foolish to say that we're going to wait that we'll have another pandemic when we're all gone and we've got the next population of human beings inhabiting the earth.
So we're not going to. We mustn't sit back and breathe easy.
We need to think about preparedness, but we also need to cope with this pandemic, which is still roaring. I looked at the paper this morning, and there were hundreds of thousands of new cases yesterday in the world.
Right.
And about 18,000 new cases in the U.S. And I still come back to a clipping that I tore out of the paper on March 3rd, 2020, saying that there had been 100 cases in the U.S. and 6 deaths. And that was an alarming sound. Yeah. Now, if we were told tomorrow that there were only 100 cases and 6 deaths, we would think this is great. So we've become climatized to the extent of the damage.
So what do we need to achieve? What degree of vaccination here and worldwide
so there are two big issues. One is overcoming vaccine hesitancy in parts of the world that have vaccine. And that's a complicated issue which we could talk about for hours because no one fully understands the multiple reasons why people are hesitant about taking these vaccines. But that's a deeper issue here having to do with a certain either uncertainty about or hostility toward science that I find very troubling, and probably linked to educational practices, to politics, to a number of factors.
But you're saying if we don't overcome vaccine hesitancy,
that's number one.
we're not going to get to...
Number two is making enough vaccine and delivering it to poor countries. I was embarrassed by the G7 announcement of a willingness to provide something like 870 million doses of a vaccine. But what we need is 11 billion doses of vaccine if we're going to vaccinate everybody in a secure way. And there's no reason why we shouldn't. Vaccine hesitancy stands in our way. But in most countries, the problem, in Africa, less than a couple of percent of the population have been vaccinated. Even until recently in Japan, where they were going to have the Olympics, only 10% were vaccinated.
Really?
And in places like India, it's 10% or less. And we do have the resources in this world. And we know now we've passed trillion-dollar bills to rescue us from something that we could protect ourselves against with a few $billion. And yet people are not coming up with the levels of money that they should be providing to get the manufacturing forces to be properly compensated for the effort they make and to provide vaccine for everybody. I mean, is there no sense of community left in this world?
So I sense a call. Yeah, thank you. But I sense a call for things to happen as opposed to confidence that they will. It's still quite uncertain from what you see.
Right now, I think it's certain that things are not going to happen in the pace that they need to happen. And it's not just the vaccine. It's also developing the technologies that allow us to know when things are going very wrong. Testing, sequencing to look for viral variants, knowing that there are viral variants that pose severe risks. I've just taken on a job at the World Health Organization running a science council. And one of the things we're being asked by the Director General, Dr. Tedros, to do is to scan the horizon for technologies that we need to put in place throughout the world. And there's nothing more important at the moment than taking the simple science, now a simple science of sequencing genomes and sequencing not the human genome, but the viral genome.
Right, exactly.
And thousands of sites around the world so that we are aware of when a variant that poses a big risk emerges.
Okay, so I'm going to mix things up with you, if I may. A little bit of a surprise. I'm going to call this the lightning round.
Okay.
And I have a series of questions, and the answers really are just of the form, do you expect this to occur in the near future, the far future, or possibly never? I'm about to tell you.
Okay.
So,
all right.
Will these things happen in the near future, the far future, or possibly never? Blood tests for main types of cancer.
Not the near future, but it'll happen.
It'll happen. Effective cures for the main types of cancer.
It'll happen.
Near, far?
Well, we already have cures for, cures are where we need to define. I'm undermining the lightning round.
All right, let's move on.
But effective treatment is different from cure.
Question. Okay.
Your cure is hard to define.
Bad question. Elimination of COVID-19. Elimination.
Probably never.
Of COVID-19. Effective treatments for dementia or other neurodegenerative diseases.
I hope. I don't see the route to doing it, but that doesn't mean anything. I mean, I didn't see the route to understanding cancer 50 years ago either.
So a cure for AIDS that doesn't involve a lifetime of heavy drug treatment.
I think that is the next 20 years.
Mainstream adoption of gene editing human embryos to eliminate single gene mutation diseases.
We could do that now, but we shouldn't, and hopefully won't. But there are ways to edit single gene, edit single genes that cause disease without changing the germline, without using embryos. And that is happening in the case of certain blood diseases as we speak.
Okay, very, very important distinction.
Not widespread.
Not a good lightning round question. Finally, last lightning round question. Gene editing human embryos to enhance traits that the parents think are desirable, such as physique or intellect.
Well, intellect's a lot harder than gender. I think...
Near, far, or never. Or shouldn't even think about it.
I'd like to think we won't think about it, but we do think we're thinking about it now. So we're so far away from it that it's really very difficult to speculate.
Okay, let's turn to public service for just a couple of minutes,
Sure.
and then I'll open it up to questions. So libraries are obviously important to all of us. What is the Public Library of Science, which you helped found, and why is it important to the matters we've been discussing?
Thank you. So about a little over 20 years ago, when I was still director of NIH, I was introduced to a very simple concept by colleagues in physics who were putting, and you probably know about this, Bert, mathematics, physics, computational science, all people in those professions were agreed that they would take their unrefereed work and simply post it as what we call a preprint before formal publication, so that anybody who had access to the internet could read it and comment on it. And that wasn't adopted initially in biology, but in consultation with some very lively friends, we decided it was time to push for the scientific literature to be much more open, and for things to be posted more quickly, and for the work that is paid for by the US government to become accessible to all citizens without being hidden behind the paywalls of very high subscription rates. We had a way to do that through something called PubMed at the NIH, an online library of titles and abstracts of all published work, simply by expanding what would be in that library to the full text of the articles. There was much resistance from the publishing houses that annually make billions by taxing the scientific community to publish their work. And when we found that the publishers were not going to cooperate, a few of us built an organization called Public Library of Science, which was initially an advocacy group to get much more enthusiasm for making science a more open avenue of communication. When that didn't work, we started our own publishing house, which is called Public Library of Science. Still publishes actually quite a few journals.
So is this accelerating news about code research?
It's not so much accelerating as making it
Yeah.
open. And it depends on a different business plan. Instead of having people subscribe to the journal, it depends on the authors, and actually it's the funding agencies that pay for the author's research to provide the money that allows the publishing house to do its work. So then the articles go online and they're open access to anybody. And they're also in the public library that the NIH maintains. And that has changed, that and the introduction of these unrefereed preprints has changed the approach that people take to the communication of science. And that's been dramatically accelerated by COVID-19, because everybody gets behind the idea that we need to know today what we learned yesterday. Can't wait for 6 months or a year.
The news cycle is faster in science now, too.
And people understand the importance. But, I would argue that, yes, COVID is very important, but why shouldn't we know what my neighbor down the hall discovered in his cancer research laboratory or his Alzheimer's studies just as quickly?
Right. Well, we don't have that in law yet, I have to say. So there's room for improvement there. We talked about diversity just a few minutes ago. What about diversifying the ranks of scientists?
Well, this is a huge problem that we've been letting fester for all too long. And I have to say, most of us who recognize that large components of our society, actually, especially Blacks, Hispanics, other people of color, indigenous people, have not occupied the ranks of all sciences, not just, it's math and physics and computer science and engineering as well as biological sciences. And we've tended to say, well, these people are not in the pipeline. We can't do anything about hiring people because people aren't there. Well, I think there are a lot of things wrong with that argument, and we simply have to change, and it's going to be expensive to do so, to change the impediments to getting a full repertoire of ethnicities into science. I know our country is becoming a very different world demographically. Very soon, the majority ethnicity will no longer be a majority, be less than 50%. And it's already true that if you look at people who are under the age of 25, that whites are not a majority. And science is going to suffer if we don't open it up. So
final question: What three other things does biomedical research need to thrive going forward?
Well, it always needs money. I mean, there's no doubt about that. And I think that's not been. It's only been a problem when the demand for money exceeds its availability. In other words, if one of the things that you praised me for in describing my time at the NIH as director was my ability to increase the size of the NIH budget. That, in turn, drove a dramatic increase in the number of people signing up to become trained to be biomedical scientists. And that created a demand that exceeded the size of the budget, especially when the rapid increase in the budget petered out over the next several years, and the inflation eroded those increases, and we had a massive number of new people, but a declining number of dollars in what we call real dollars. So
So I think
money.
we're still on item one.
Number
Yes.
two is talent, and that means attracting everybody from whatever ethnicity or socioeconomic group or country that you emerge from, and get them interested in these really fascinating problems that have such a large effect on human health, to recognize that science, and especially the biomedical sciences, which I have to say are less and less biological every day. The role of computers and physics and chemistry in the study of human health has become more and more apparent every day. So virtually every discipline contributes to the sciences that affect health. So those are two. The third is probably has something to do with the support of the culture for the kind of thing we do. That is recognition of its importance. It's the role that science can have on our excitement about life, our ability to improve life. We're not here that long, but we can do something to try to improve the way future human beings live. And if that level of excitement about what science can do for humanity is out there, and unlike the current desperation and conspiracy theories and suggestions that if anything bad happens, it's because of some plot that some scientists or Bill Gates have to undermine the condition of humanity. Subterfuge.
Well, I think you've done a great job of building some enthusiasm and support in this area. So thank you. So now we have about 15 minutes for Dr. Varmus to take questions about the future of medicine and biomedical research. I'm just going to ask you one thing. Please refrain from mentioning your own medical conditions or your loved ones. Specific cases. Our focus is on research and the general future here. So raise your hand if you'd like to ask a question, and the microphone will be brought to you. Yes, hi, please.
Thank you, Dr. Varmus. It was really an honor to hear you speak. I was so excited I got here early for a front row seat. I'm a veterinarian, and I also sit on a scientific board whose mission is to study pandemics and how to prevent them.
Sorry, your mission is what?
To study the prevention of pandemics and how to predict them. And my worlds combine as a veterinarian and on this board because most emerging diseases have their origin in wildlife. So
Your board is not EcoHealth, is it?
it is EcoHealth Alliance.
Is it?
Yes. And regards from Peter Daszak.
Thank you.
So COVID-19 has made my job much easier in explaining what a pandemic is and why we want to prevent them. And but my question to you is, what we're very good at is reacting to what's happening. But we know from previous pandemics, and maybe they didn't reach, global state, and maybe they didn't cause, enormous economic devastation, but many, many people died. And sometimes it was just the luck of where the carriers came into the United States, what happened here. So with your NIH hat on, how do we work to prevent these diseases rather than spending trillions of dollars cleaning them up? Like, we know things like deforestation causes. We know it's...
Let's
So
just go to that.
Yeah, so I think there are some answers to the questions. One word that I would emphasize, and I'm sure you'd agree with this, is surveillance. And we have much better tools for surveillance than we ever had before, that we now know we can actually look at sewage from a city. And you're familiar with this, but for the rest of the audience may be unfamiliar, that the idea that you can take the effluent from a large community and look at it for the appearance of unexpected organisms that may be pathogenic, may be able to cause disease. I think it'd take a long time to get the world to agree to do these things. But I think where I've been asked about this kind of thing, my own view is to begin at the level of major cities. That a city like New York or London or Tokyo has the ability to begin to monitor everything in its environment, and to do so using genetic tools that are very powerful. And what I'd like to see happen is have cities around the world build a necklace of surveillance centers where people are coming and going, where there are large numbers of people of many different ethnicities, where they have the academic institutions that can help build these resources. And I'm hopeful that we can actually do more as a result of not just because this pandemic has been so deleterious to human health, but also because we do have new scientific tools that are really powerful. I mean, your organization, EcoHealth, uses them, and they are. It's not a matter of looking for one or two pathological organisms. It's being able to look for almost anything that might appear. Well, is this going to be perfect? No, but it's a hell of a lot better than what we're doing right now. But there is something that we have to seize on at the moment, and that is the recognition, as you say, that pandemics are incredibly expensive, and they are recurrent, and we have to make some investments. One of the things that makes it hard to get started in this country is that every time we've had an epidemic or a pandemic in this country, there is interest in doing something about surveillance and other kinds of protection for a short time. And that interest wanes because we go for a year or two and nothing else has happened. And we have undermined interest in public health. And I think there are a lot of reasons for that. You can see it when you compare the vitality of medical schools to the difficulties that schools of public health have in raising money and attracting people to get engaged with public health. And hopefully there's going to be enough of a change in the mindset of people as a result of this pandemic to do something about it. But we desperately need a COVID commission that has the support of people, doesn't appear to be a sectarian or a partisan investigation. It doesn't have to resolve the question of where the SARS-CoV-2 came from. Worth looking at that, but we have a lot of other questions to ask about the nature of preparedness, not just surveillance, but having stockpiles, national stockpiles of equipment, having scientific platforms that can that can generate vaccines in less than a year, that can generate drugs and oversee their regulation in an appropriate way. So there are a lot of things that need to be done. And my hope is that the pandemic, for all of its ill effects, has also been a wake-up call.
Okay, another question? We've got another in the front here, and we'll go to the back afterwards, okay?
So I live in a slightly different world. I'm a medical oncologist, and for the last 33 years, one of the concerns that I have is our current paradigm for cancer treatment is, like you said, at the form of the metastatic disease. The search for the primordial cancer cell, Dr. Raza, who runs the myelodysplasia laboratory at Columbia and is one of my compatriots, has saved 50,000 bone marrow samples because she is looking for cells that uniquely start to have a single mutation, the ones that we know about in CML and some of the easier things, so that we know, as you said, cancer can have 50, 100 mutations, but somewhere along the way, the cell starts. We have some technology, we have PCR, we have sequencing. Where do you see that all coming in a world where we don't have COVID every day?
Well, the problem that we face is that, yes, you can find mutations early, but you don't know if they equate to disease. And you don't want to be giving people who are never going to have frank cancer the kinds of drugs that you and I know about. So one of the things that is a startling scientific development over the last just couple of years that displays this concern is that every time a cell divides, it acquires mutations. It's been recently shown that when you take an egg and fertilize it in a test tube, as you would in vitro fertilization clinics, when that first cell divides, the two daughter cells have differences because of mutations that occur during the copying of the DNA and cell division. When are you going to say that a mutation is cancer causing? We know now that if we sample the DNA of people in this room from their normal blood, that perhaps 20 or 30%, maybe more, would have mutations in genes that we know can contribute to cancer. But we're not going to start treating those people for cancer, and nor do 20 or 30% of us want to be under monthly surveillance to see whether we get the disease. So there's a balance between our powerful capacity to identify mutations and even to recognize them as potentially worrisome, and our ability to say a diagnosis is being made and some kind of treatment should ensue. So I'm very sympathetic with what you're raising, and I'm aware of Dr. Raza's remarkable collection. But and I am hopeful, as I indicated in the lightning round, that we will have blood-based so-called liquid biopsies that allow us to make diagnoses of cancer at a much earlier stage. But it is still hard to know when you've crossed that boundary from having mutations to having cancer.
So there were one or two people in the back there. Can we get the first one? Yes. Thank you.
Thank you very much. I wanted to talk for a moment or ask you about the archive system, the bioarchives and so forth. I think one of the remarkable things, maybe the one silver lining about this whole pandemic, has been the level of cooperation among laboratories all over the world, sharing data, sharing resources, et cetera, et cetera. And I wonder what it is you think we can do to continue that post-COVID and not slip back into the culture of competition that had so overtaken much of science.
No, I think that's an important question, but I would, just for the sake of my enterprise, say a couple of things about your framing of the question. First of all, I think a lot of scientists are very cooperative and collegial before a pandemic. And my own life in science has been built heavily on collaboration, not just among people who work in the same lab, but among various labs that could, in principle, have been competing. And I know I've found this a wonderful world to work in, and the there are bad apples, of course, but it's not a cutthroat world, and it's one that does recognize the virtue of there's virtue to competition, there's virtue to collaboration, and I think it's a reasonable world. But I do agree that we have highlighted the collaborative instinct and shown its virtue during the pandemic. It's not been perfect, though. One of the things that I'm wrestling with at the moment, because I've been asked, as I mentioned, by the World Health Organization to think about how we put the tools of certain new scientific disciplines like genomics, the analysis of the sequence of viral genomes and human genomes into put those technologies in place throughout the world for the benefit of human health. And there are lots of reasons to believe we can, not just in the realm of a pandemic or cancer, but in lots of other diseases, undiagnosed and hereditary diseases, for example. We can make tremendous advances by obtaining that information. But even in the pandemic, it's not so easy to get access to all the information that's being generated by people doing the sequencing. People who've done it don't want to lose the chance to get credit for it. There are countries that are worried about getting their reward for contributing to data access to databases that provide information to everybody. You may recall some years ago in Indonesia, they were collecting a lot of sequences from new influenza viruses that were being identified, but there was some deep reluctance to share the sequencing information because the claim was, we'll put the information in there, vaccines will be developed, rich countries will get them, we won't. So they were using data access as a leverage point for assurance that if they contribute to knowledge, the benefits of that knowledge will be shared. Those are all legitimate concerns, but to me, don't rise to the level that it provides a justification for limitations on data access. I don't know the solution to this. I do know that this week WHO is having a meeting to try to discuss how best to get a much more complete sharing of information about the viruses causing the current pandemic, because we don't have the kind of access we should have to all of the sequences that would help us define when significant new variants are emerging. So, I've just complicated the problem further, but I think you're right in drawing attention to the fact that, yes, people are more understanding of the need to provide access to information, but it's not always happening.
Well, I think with that, I think we'll have to cut the questions off. There will be a reception afterwards, and I think a chance to talk to Dr. Varmus then. Let me thank Ben and Donna Rosen, especially for sponsoring this event, and the Kent Memorial Library. And of course, Harold Varmus. Thank you so much, Harold.
Thank you.